Our final speaker for this section is Karyn McFadden. Karyn is a postdoctoral fellow in Mika Luftig's lab. She got her Ph.D. in biochemistry from Drexel under Erwin Chaikin, characterizing new inhibitors of HIV. And her talk today is entitled EBV-Mediated B-Cell Transformation is Suppressed by Oncogene-Induced Senescence through the DNA Damage Response. Karyn?
Thank you, everybody, for having me speak today. HIV infection is, there is an increased risk of malignancies in patients that are infected with HIV, such as Kaposi sarcoma or a number of B-cell lymphomas. The advent of antiretroviral therapy has substantially reduced the risk of developing a number of these malignancies, such as Kaposi sarcoma. However, the risk of developing lymphomas remains elevated, and this is probably due to the immunosuppressed status of the patient. However, there are other underlying cofactors, such as co-infection with KSHV or EBV, the focus of my talk today. And EBV infection has been found in a large number of B-cell lymphomas to various degrees. Now, to take you through a very brief background on EBV, it is a gamma herpes virus that is spread through salivary contact. EBV initially infects naive resting B cells, and then the infected B cells undergo the normal B cell maturation process. And the reason that I'm bringing up this slide, too short to be up here, is that EBV-associated tumors reflect their B cell ontogeny, in that EBV goes through 3 different latency stages, and latency 3, which is characterized by the expression of the full complement of EBV latency proteins, is seen in AIDS diffuse large B-cell lymphomas, whereas Hodgkin's lymphoma is characterized by latency 2, which has a more restricted expression of EBV proteins, and Burkitt's lymphoma is latency 1. Now, EBV is a ubiquitous virus. It is pretty much everybody in this room is infected with it. However, most of us are able to suppress EBV-induced lymphomas. And this is primarily due to having a strong immune system. However, other factors are at play. And this is partially observed many years ago, in that you can infect EBV in culture, primary B cells, to an extent where you get about 100% infection, and only about 1 to 10% of the B cells will end up transforming and becoming LCLs or lymphoblastic cell lines. And this implies that there is an innate barrier to EBV-mediated transformation. And the focus of our Mika's laboratory is to understand what this barrier is. One possibility is the DNA damage response, and I'm showing this in cartoon form here, in a grossly oversimplified, but we know that if you overexpress an oncogene, you can induce aberrant proliferation. This aberrant proliferation induces replicative stress, such as through the depletion of nucleotides, and this can form double-stranded breaks, particularly at fragile sites. Normally, the DNA damage response would then be triggered, and this is a signal transduction cascade, initially through ATM or ATR, which activates through phosphorylation, P53 among a number of other proteins, and this leads to apoptosis or permanent cell cycle arrest or senescence. However, in the absence of the DNA damage response, you could have genomic instability. This could result in the fragmentation of chromosomes or translocations such as seen in Burkitt's lymphoma. And with additional mutations, you can end up developing cancer. And it's known, if you look at pre-malignant nevi, that they have a number of markers of senescence, implying that this is a true way that the cells are able to suppress malignancies. So the first question that Mika wanted to address was whether or not EBV infection could recapitulate oncogene overexpression. Did you have this period of hyperproliferation and the DNA damage response controlling it? And this was work done by Pavel Nikitin and Chris Yan. And in these experiments, we isolate PBMCs from human donors, infect them with EBV at a high titer, and we also label them with a proliferation tracking dye, so we could see the number of cells that have proliferated once, twice, etc. And if you look at division number over time, what we found is that there is an initial burst of hyperproliferation after about 3 days in which the cells will proliferate up to 3 times a day. This eventually attenuates to levels that are more like what you would see in LCLs or lymphoblastic cell lines. Since we're able to track the proliferation of the cells, we're able to isolate cells that have either not proliferated, proliferated once, twice, etc., and assay them for markers of the DNA damage response. And that is shown here through an immunofluorescence study in which we can see either EBNA-LP, which is a marker for EBV infection, or H2AX as a putative marker for the DNA damage response. Let's see if I can do it this way. And what we can see is that we have a population of cells that have never divided, but they are infected with EBV. Cells that have divided once or twice have this elevated markers of the DNA damage response. This is also seen in cells that have divided up to 4 times, and this is ultimately attenuated over time. And I'm not going to go on about this too much since this is published work, but Pavel then went on to show that if you inhibit the DNA damage response, you have an increase in viral transformation, implying that the DNA damage response is a true method that the cells can use to attenuate EBV-mediated transformation. I then wanted to take this a step further, and just to give you a quick experimental design, what I do is I also isolate PBMCs from patients and EBV infect them and label them with a violet stain. After the initial burst of proliferation around day 3.5, 4, I label them with the second stain, CFAC. So in this population, you have cells that have either not divided or have divided 1 through 5 different times. And then we can track this over time. I usually do my analysis toward the end of the hyperproliferative period around day 7. And what we can see is that we have a population of cells that have either become infected and never divided, some that go through this proliferative burst and then arrest, and cells that maintain their proliferative status. So the first thing we wanted to know was whether this was an artifact of the cell culturing conditions with the depletion of nutrients, or whether these cells were truly arrested. So I isolated the different populations, and then just looking at the number of cells over time, we could see that our proliferating population continues to proliferate happily in culture, and these ultimately become immortalized cell lines. Whereas my arrested and non-proliferating populations, I can't induce them to proliferate. And I looked at this up to 2 weeks, but we've seen this a number of times. And this is 3 independent donors. And this was also seen in Ki-67 staining. What I could also do is, since I have these 2 proliferation stains, is look at cells that had divided once, twice, etc., and see which of these populations proliferate versus go on to arrest. And what I noticed is that the arrested population was enriched in this PD1 to 4 population. And of note, this is the population where we originally saw the elevated markers of the DNA damage response. So then just isolating proliferating cells or arrested cells, once again looking at H2AX as a marker of the DNA damage response, we could see that the arrested population is elevated for H2AX. So this implies that after EBV infection, the cells go through a period of hyperproliferation. There is then a DNA damage response. This DNA damage response can then arrest a large population of the cells, and this is one of the factors that is limiting EBV-mediated transformation. We also wanted to know where in the cell cycle this arrest occurred. Through work that I don't have time to show, we know that EBV protects the cells against apoptosis, so that this was not through an apoptosis and more likely through a cell arrest. And in this study, and this was work done with Pavel Nikitin again, we infected B cells with EBV and then inhibited them with a chemical inhibitor of the DNA damage response, and looked at on day 7 for progression through the cell cycle through a BRDU assay. And what we see is in our day 7 cells, there is a large increase in the number of cells entering into S phase. And this implies that the DNA damage response is slowing EBV infection by inducing a G1 S phase checkpoint. Now, if we look at LCLs, and these are the same cells, but they have gone on through day 35, they don't have markers of the DNA damage response. There is no difference in growth phenotype in response to CHEK2, and also there is no difference in moving into S phase. And just also taking this a step further, I looked at my arrested versus proliferating population, and I'm only showing a small subset of what we've done here, but we looked at some stereotypical markers of cell cycle progression, such as P21 or P16, and what you can see is that there is an increase in P16 and 21 in the arrested population, and markers of genes that are important for progression into cell cycle proliferation are reduced. So I just want to end on a kind of a global summary. What our lab is interested in is understanding how, what the innate responses that can prevent EBV-infected cells from becoming immortalized LCLs or malignancies. And what we found is that the DNA damage response is triggered in response to this initial burst of hyperproliferation, that this induces cells that become arrested through a G1S phase checkpoint. And current work in our lab is focused on exploring what are the triggers, what is causing this DNA damage response, is it the replicative stress or is it mediated through telomere dysfunction, and also identifying the downstream pathways. And I'm going to end on this in the interest of time and everyone getting their lunch, but I wanted to thank in particular my postdoc mentor, Mika Luftig, who always keeps things interesting, as well as funding through the training grant with Dr. Hicks, as well as CFAR funding. Thank you.
Any questions?
I'm sorry? What about Burkitt's lymphoma?
Mm-hmm.
How contrast what you've seen, or compare contrast what you've seen here to what may be going on in Burkitt's lymphoma?
Well, Burkitt's lymphoma is a latency one, so there's less gene expression there. But what you can say, Burkitt's lymphoma is characterized by the IgMYC translocation. And what you can say is that there could be the accumulation of some of these fragile sites mutations that lead up to the culmination where if it's not repaired and it's getting passed on over time. You could also say in the Burkitt's lymphoma model that there is a contributing factor, obviously, of EBV infection, and it could be that in HIV or in malaria, where it's even more seen, you have EBV preventing apoptosis and also mediating some of this hyperproliferation. And it also, EBV, I didn't get a chance to go into the EBV proteins, but one of the earlier proteins turns on MYC. So if you have MYC going in hyperproliferation, any additional stimuli from HIV could induce these cells to undergo the translocation, and also just the overall lack of immune suppression of catching these cells and killing them.
Any other questions?